Yeah.
What's Gucci? I'm your host, Aquarius the Third.
And I'm your second host, Shamar.
Today we're going to spill on some real ish, the process of protein synthesis.
Protein synthesis involves two main stages, transcription and translation. By God Tupac himself, I'm about to go ham on this transcription. That is just broken down further into three stages: initiation, elongation, and termination.
During initiation, DNA unwinds and an enzyme known on the streets as RNA polymerase initiates RNA synthesis. After protein synthesis just got initiation this to elongation.
Alright. In elongation, RNA polymerase runs up on the DNA and continues to bust all up in that dude's double helix, exposing about 10 to 20 DNA nucleotides at a time for pairing with RNA nucleotides. As always, nucleotides is added from the 5' to the 3'. And by the way, that prime shit reminds me of the Transformers and my OG bro, Optimus Prime.
Oh yeah, Optimus Prime, that guy kudo.
Anyways, let's keep this into termination.
Termination is different in bacteria and eukaryotes. In bacteria, the transcribed terminator, that's right, the terminator, yeah, functions as the termination signal, causing the polymerase to detach and release the transcript, which requires no further modification, unlike my caddy, throwing some 24s and spinning some rims. They spinning, player, they spinning.
Alright. In eukaryotes, RNA is modified after the transcription. Each end of the pre-mRNA molecule is modified. At the 5' end receives a 5' cap, but not like the caps that not those caps. Don't any of you dudes be running up on me now.
Don't do it, don't do it.
Sorry, this cap in protein synthesis is a modified form of guanine nucleotide. On the 3' end, a poly-A tail is added, which has 550 to 250 more adenine nucleotides. The 5' cap and the poly-A tail then bust they bust the mature RNA out of the nucleus, just like we got Von Quick out this joint.
Once transcription is complete, translation can begin. In translation, a cell reads. I wish I could read.
Anyways, a genetic message and builds polypeptide accordingly. The message is a series of codons along with the mRNA and the transfer RNA, or as the homies call it, tRNA. That translates. tRNA runs amino acids from the cytoplasmic pool of amino acids to a growing polypeptide starting at the codon, an OG known as AUG.
Each tRNA molecule translates a particular mRNA codon into a particular amino acid. This occurs in the his house of a ribosome. Ribosomes hook up tRNA anticodons with mRNA codons so well it makes my pimp daddy self-proud as I'm sorry. A ribosome got a large subunit and a small subunit, each made up of proteins and one or more ribosomal RNA or RNAs. Each ribosome got 3 binding sites for tRNA. The P site holds the tRNA carrying the growing polypeptide chain, but that's not all we grow down her in the hood.
We grow down potatoes growing. The A site holds the tRNA carrying the next amino acid to be added to the chain. The E site is where all the discharged tRNAs dip hard from the ribosome. When a tRNA molecule runs up the ribosome, it's gotta have some good.
It better have that.
A specific amino acid on one end. At the other end, this tRNA is a nucleotide triple called the anticodon, which got the base pairs.
You do got base pairs.
With the complementary codon on the mRNA. Codon by codon, the genetic message is translated as tRNA swerving through the ribosomes and make drive-bys dropping amino acids in order they prescribed. Now because they got the PhD ballin'.
I know that the ribosome then joins the amino acids into a chain.
2 chains.
This continues until a stop codon is reached. This play signals for the translation of the mRNA to stop. The polypeptide chains is now some thug player proteins.
Got real proteins. Real proteins.
Hey. No. This has been Delquarius the Third.
And Shema, keeping it real. Thank you for your time. Please stay in school. Pick your book up.
You ain't my mama, you can't tell me what to do. During initiation, DNA unwinds in an enzyme known on the streets as RNA polymerase. RNA polymerase runs on the DNA. Oh wait, whoops, that's wrong.
She ain't approved, so you know they had to bruise them. That's the way they do, man, they swear they so gangsta.